Hello, everyone, and thank you for tuning into my talk. My name is Orion McCarthy, and I'm a second-year PhD student in Dr. Jennifer Smith's lab at the Scripps Institution of Oceanography. And today I'm going to be talking about the three-dimensional structure of coral reefs and how we can learn something about coral reef ecology by measuring this structure in a multi-scale way. But before I get into the specifics of coral reef ecology or multiple scales, I want to take a moment in general to talk about the ecological relevance of structural complexity. And so the diversity, size, and type of structures in an ecosystem creates niches and microhabitats for different organisms to exploit or use as refuge. And in this way, ecosystems with high structural complexity can support high levels of biodiversity. And coral reefs are no exception to this and owe much of their biodiversity to this structure. And superficially here, you can see the difference between a reef with high structural complexity and one with less structural complexity. And you can imagine how those two reefs are capable of supporting different amounts of biodiversity. Now, reef-building corals are the main source of biotic structure on reefs, but because corals are living, the structure they create changes over time and is vulnerable to disturbance. Coral cover is expected to decline throughout the tropics under climate change, and thus we also expect to see a loss of structure in these habitats. And that could have implications for many other organisms that live on coral reefs. And to show you why, it's helpful to consider multiple scales. This conceptual figure shows a number of different structures that exist on coral reefs, both biotic and abiotic, across a range of scales. And organisms relate to this structure as a function of their body size. And this intuitively makes sense. A hermit crab won't interact with the substrate in the same way as a large reef fish would. Furthermore, if there are a lot of structures in a certain size range, we would expect to see a lot of organisms with a body size that matches those structures. And this is known as the textural discontinuity hypothesis and was first posed to address the size distribution of birds and mammals in various habitats. But it has also been found to apply to coral reef fish as well. And here are some pictures illustrating this point. You can see small organisms utilizing the fine scale structure of coral branches, while slightly larger fish may use the overall shape of that same coral colony for shelter. And on larger scales, fish also depend on the underlying topography of the reef for habitat. So by measuring structural complexity at multiple scales, we can begin to tease apart these drivers to determine what makes a reef complex, and then make conclusions about what types of organisms that reef may be able to support. So that leads me to my research questions. My overall aim in this presentation is to demonstrate that scale provides context and can enhance our ability to reach ecologically relevant conclusions about a habitat's structure and change over time. But first, I want to take a moment to explain how reef structure has historically been measured and how we're harnessing new technology to measure structural complexity in a multi-scale way. And so to start with that, here's a picture of me and some colleagues measuring structural complexity on a reef in Hawaii. And one of the tools we use to measure structural complexity in the field is called a profile gauge. It functions similarly to a toy you might have had growing up, the one with a bunch of pins in it that you can put on your hand or your face to get a general outline of that surface. The profile gauge works the same way to measure this orange line, the contour surface of the reef. And so taking a ratio of this contour distance to the straight line distance of the profile gauge allows you to quantify the complexity of the reef. And that ratio is called linear rugosity. So a higher number indicates a more complex surface, and it's lower bounded by 1, which would be a completely flat surface. And although linear rugosity is the most widely used metric of structural complexity, measuring it in the field can be challenging due to the time constraints of a scuba dive and the precision of these measurements in situ. And these constraints make it challenging to measure the same habitat more than once at different scales. And for that reason, the majority of studies on coral reef structure have been single scale. However, this field has rapidly changed in recent years with the rise of 3D modeling using a technique called structure from motion. And structure from motion refers to the ability to create a model of a surface using the overlap between photos. And the Smith and Sandin labs at Scripps have been using structure from motion to survey reefs from around the world. And instead of taking profile gauges with us into the field, we take cameras, and we take thousands of pictures covering an area of about 100 meters squared, swimming in a gridded pattern to ensure a lot of overlap between those photos. And then when we get back to the lab, we stitch those photos together into a 3D model like this one, which we analyze using a software called VisCore, which was developed at UC San Diego. And this is great because it basically allows us to take the reef home with us. And we can use it to draw virtual profile gauges across the reef's surface. And we can draw as many profile gauges as we want and place them in the exact same location over time if we have models of the same place in multiple years. So this is already a major improvement over in situ methods for measuring ergosity. And this is just an example of what those measurements look like in BizCore. Each of these orange lines is a virtual profile gauge draped across the surface of a 3D reef model. And so each virtual profile gauge consists of a series of points spaced at regular intervals along a line. And by connecting those points, you can get the contour distance across the reef and then calculate rugosity. But unlike in the field, you can also change the spacing of those points to simulate different scales of measurement. And here you can see the rugosity value you get actually changes depending on the scale of measurement, even though the underlying surface we're measuring doesn't change. And we can use that multi-scale rugosity data to tell us something about the size of structures on a reef. So let's look at the rate of change in rugosity between the 1 centimeter and 2 centimeter scales. That decrease in rugosity here is entirely attributable to structures between 1 and 2 centimeters in size. And this change in rugosity between 2 and 4 centimeters is due to structures within that size range. So by quantifying the rate of change in rugosity between each scale, we can partition structural complexity into a series of size bins. And the metric we use to do this is called fractal dimension. And so a high fractal dimension tells us that there are a lot of structures in a certain size range. And in an ecological sense, it also allows us to quantify the amount of habitat available to organisms of different sizes. And so for my first year research project at Scripps, I used fractal dimension to distinguish between biotic and abiotic reef structure. And so as these conceptual figures show, I found that biotic structures, aka coral colonies, are primarily responsible for high fractal dimension at the scale of several centimeters, smaller scales. While abiotic structures such as rocks, boulders, or overall reef geomorphology created high fractal dimension at the scale of several meters. And I'm not going to go into more detail here. The main point I want you to take away from this is that cross-scale patterns of fractal dimension can help us to identify the underlying processes that create structure on reefs. Since my first year presentation last year, I've focused on using this fractal approach to distinguish between different types of corals in terms of the structure they create. And to explain my interest in coral-driven complexity, I'll draw your attention to these pictures here. Each of these reefs is from the Hawaiian Islands and has a good amount of coral cover, but the amount and type of structure that those corals create differs depending on the genus and growth form. And because of these structural differences, it's likely that these reefs provide different types of habitat and support different organisms. So to characterize these differences, we measured the rugosity of different types of corals at multiple scales and calculated the fractal dimension at 6 scales. So you can see the scale intervals here on the x-axis, the fractal dimension here on the y. And we identified sections of each reef model with uniform benthic cover so that we could control for coral type. And the 2 genera we focused on were Porites and Montipora, which were the most abundant in the Hawaiian Islands. And here you can see that the amount of structure created by Porites corals, for example, depended on the growth form, with branching corals creating more structure than encrusting or massive Porites at most scales. And we observed a similar pattern for Montipora, which you can see here. We also found that skeletons left behind after a coral dies also provide some level of structure, actually comparable to that of an encrusting coral. And this is important to note since coral skeletons may persist for several years after a coral colony dies, say following a bleaching event. So this implies that the amount of structure you see on a reef after a disturbance like that, there might be some residual structure left over from dead coral colonies. And so it's useful to be able to distinguish between coral structure at a colony scale, but as this screenshot from one of our models demonstrates, reefs are characterized by different types of corals. So it's also beneficial to look at the community scale to see if we detect the same patterns. And to do this, we surveyed 10 by 10 meter sites from the Hawaiian Islands and classified those into 4 different coral communities. And to do this, we grouped sites based on the most abundant type of coral at those sites, and then grouped those sites based on community type and compared the mean fractal dimension of those groups across scales. And we found significant differences in structure between these types of communities, with branching parietes reefs exhibiting the most structural complexity. For the most part, the community scale patterns mirror patterns from the colony scale, although one exception is that reefs with high abundance of encrusting montipora have higher fractal curves than expected from colony scale data. And this may be due to the common co-occurrence of encrusting montipora with branching parietes, whereas reefs dominated by branching montipora, for example, were less diverse. It's also important to note that reefs with the highest coral cover, the branching montipora reefs, did not have the highest fractal dimension. Here we found actually that communities characterized by intermediate amounts of coral cover had higher fractal dimension values. And this suggests that structural complexity is not simply a function of coral cover, but that coral genus and growth form also play a role. And so we demonstrated that we can parse out the structural signature of different coral colonies and communities, and this suggests that we can use this method to track change in structural complexity that may result from changes in community composition. For example, if one type of coral gets the upper hand as the climate changes. And so on that note, let's transition to my final question, which focuses on using fractal dimension to document multi-scale change in structural complexity over time. And for this part of the study, I'm going to focus on reefs from the islands of Molokai and Maui, which our lab surveyed in both 2017 and 2019. And this map shows how coral cover changed at these sites over the course of those 2 years. And while most sites had fairly stable coral cover, this one site here on Maui had a pretty large drop of nearly 12% in its coral cover, which is pretty large considering it represents a loss of about 1/3 of that site's coral cover over a 2-year period. It's also worth noting that Maui has a much higher human population than Molokai, and its reefs tend to be exposed to more human impacts, such as nutrient runoff and overfishing. And so these graphs show the fractal signature of our sites from Molokai. And these figures are similar to those that I showed you before, and I don't want you to get overwhelmed by the sheer number. Just in terms of interpreting them, the scale intervals are here on the x-axis and the fractal dimension on the y, and each one represents a different site. And the beige line represents that site's fractal dimension in 2017, and the blue in 2019. And the rugosity values are here in the corner for context, and they're taken from the 0.5 centimeter scale interval. And for the most part, we can see that the reef structure was remarkably stable on Molokai over this 2-year period, with 4 out of the 6 sites barely changing at all. And we can pair this data, then 2 sites changed at smaller scales only. And we can pair this data with changing coral cover, focusing on the types of corals that we've previously discussed. We've got branching and encrusting montipora, and then branching and massive parietes. We can see that the 2 sites that had an increase in their complexity also had increases in branching coral cover, which suggests that the fractal dimension change that you see here is due to incremental coral growth. Transitioning now to sites from Maui, we can see that changes in reef structure were more pronounced over the study period, with more sites showing a slight decrease in complexity at small scales. And for our site with a 12% decrease in coral cover, this site here, we can see that much of that change was due to loss in branching parietes. Conversely, the only site where fractal dimension increased also saw an increase in branching parietes, even though overall coral cover remained relatively constant. And this fits in with our findings from earlier slides that branching parietes is one of the more structurally complex corals on Hawaiian reefs. And so digging into that one site that saw a large change highlights the flip side of branching parietes, which could be its susceptibility to disturbance. So screenshots from Honokawai, this site that saw a large decrease in complexity, illustrates that nicely. You can see from 2017, but also from 2019. And we can see that the branching parietes was badly damaged over this 2-year period, possibly due to a storm, and that resulted in the loss of a lot of structure. So while branching corals create more structure, they're also more delicate than massive or encrusting corals. It may be more vulnerable to disturbance events. So to summarize my major findings, I found that scale is an important factor to consider when measuring structural complexity. And looking at how complexity changes across scales allows us to learn something new about what aspects of a reef make it complex. And these cross-scale patterns can tell us something about the type of habitat reefs can provide for organisms and can allow us to better contextualize changes in rugosity over time. Finally, I hope this study showed you how technologies such as structure for motion can be harnessed to expand the utility of existing metrics, such as linear rugosity, to allow us to learn something new that wouldn't be possible using conventional field approaches. And with that, I'd like to thank my committee, particularly my advisor, Jen Smith, everyone in the Smith and Sandin labs, my great volunteers, Elise Gonzalez and Connor Elliott, and all the support staff at Scripps, and my cohort and friends and family for supporting me thus far, and of course my funding sources.